good question. So 95, I don't think there's any particular reason except for the fact that that was the amount of time we could go backwards into and still be confident that we had a good measurement, okay? Because if you make it much shorter than that, you think about it, you're only dealing with a few millimeters of sediment. It's hard to do that very accurately, okay? So we just wanted some period of time that we felt good about that was fairly recent. And so they all tend to accumulate more. They do it at different rates in these different systems. But the other thing I wanted you to get from this figure is we don't seem to see any clear differences in terms of the clear systems versus the turbid systems in terms of how much carbon is being buried. Now, one of the problems here with this is, in response to Christine's question, so it takes a fair bit of soil in order to make a measurement, okay? And one of the problems is that some of these systems switch back and forth between clear and turbid. So it may be clear today, but it may have been turbid in the past and vice versa. So it's hard for us to say with any confidence that you know, this system is turbid and it's been turbid for a long period of time. Okay. These are data from one lake, Lake Christina, which is a very large, shallow lake in that part of the world, basically. And we know from historical records that this lake has been managed very heavily to basically switch it from turbid into clear states. And so we have good records of when it was turbid and when it was clear. And you can see it switches back and forth, but there's no clear pattern in terms of carbon burial, how that's changed, except for the fact that it is accumulating more carbon today than it has in the past. Okay. There's a little bit of a problem with interpreting it too heavily in that way because this material has also undergone more decomposition. But as I said, as soon as they go anaerobic, the decomposition rates drop off quite a bit. Okay. Another thing we've been looking at is just in the turbid and clear state, the plants are different. So microalgae presumably decompose much faster, so farther to the right, it's high decomposition rates, than the macrophytes, which are a little bit further to the left and decompose a little bit slower. Okay. So that could have an effect on storage, but as we just saw, there's really no evidence that the macrophyte dominated systems or the phytoplankton dominated systems decompose at different rates. We've also looked at the dissolved pools of organic matter in these systems, and there doesn't seem to be any systematic difference in the rate at which that material decays. So if you think about it, so a plant, most of the biomass of the plant is in the plant itself, but it's also excreting material into the water. We see a little bit of difference between the macrophyte and the clear systems in terms of the rate at which that organic matter breaks down. So the macrophyte systems tend to decompose a little bit slower than the algal-dominated systems. But these numbers don't seem to be, well, it could be significant. We just haven't looked at it that much yet. And as I said, if you look at all the systems and compare the clear, turbid, and the ones that are sort of switching back and forth, there doesn't seem to be any real difference in terms of the total amount of carbon that's buried. But there's a lot of carbon that's buried in all these systems. Okay, so both system states tend to bury a lot of organic carbon. And I know it's hard to, if you don't have experience with it, it's hard to put this in context. But these numbers are extremely high, especially if you go back and compare to some of those mean numbers I was giving for freshwater systems throughout the world. We said small lakes bury on the order of 70, and we're up around 150 in these systems. So they're about twice as much as your average small lake is bearing. We've also looked at whether or not carbon supply, or the exchange of CO2 into the plants to begin with, might be an important factor. So this is Leah Domini. I forgot to mention my collaborators on a lot of this work. Leah's one of them. Kyle Zimmer, who some of you may know at St. Thomas, is another collaborator. And Will Hobbs is the guy that did a lot of the sediment work. Okay. So we want to know basically, how does the amount of carbon that's coming into these systems through photosynthesis compare to other systems? And again, here's our diagram, kind of remind you. So this is basically what we're looking at in this process, is the exchange of CO2 and the movement of CO2 from the atmosphere into the plants themselves. Because that sort of sets the bar in terms of how much carbon they have to work with in the first place.
Was that a data sonde in the previous slide? And what sensors were you using? So the sensors we used for that are pretty standard. So dissolved oxygen, pH, conductivity, and temperature. And I'll show you some of the data, but mostly we use DO in order to calculate productivity. And we're not directly measuring CO2, so that's a little bit of a problem. But CO2 and oxygen are coupled through photosynthesis and respiration. So, and those relationships are pretty well known. So that's why we did it that way. But one of the fluxes that's not included in our photosynthesis calculations is basically material that comes in what I think of as horizontally. So it comes from the watershed. So if you have sugars or carbohydrates or proteins that are being transported across the landscape into the lake, they can still metabolize that material. They can respire it, but it's not part of our photosynthesis calculation because we're just looking at the change in dissolved oxygen over time. Okay? And one of the ways that this is important is a lot of work has shown that most lakes basically consume more oxygen than they produce, okay? And so one of the reasons for that is if you have a lot of material that's coming in from outside the watershed, you can consume more oxygen or produce more CO2 than what you're generating in terms of photosynthesis, okay? So we like to say that most lakes tend to be heterotrophic. The question is, what about these pothole systems? What are they autotrophics? In other words, do they take in more CO2 than they respire? Or are they heterotrophic? Do they breathe out more CO2 than they photosynthesize? And this is how we do it. So dissolved oxygen. So you can look at the changes in oxygen and relate that back to the CO2 that's being produced during the nighttime, and then the CO2 that's being taken out during the daytime. So the peaks and valleys on those curves. And if you look at the real data, they kind of look like this. They're a lot messier. So the temperature in these two different lakes, these are prairie pothole lakes, it can vary quite a bit in these systems because they're so shallow. And then these numbers here are the dissolved oxygen in these two systems. And so you can see, that you can see some peaks and valleys in these systems, and actually the oxygen can even go quite low in these systems for extended periods of time, probably related to mixing events that occur in these systems. But they're pretty dynamic. They're fairly simple. This is like a fairly simple part of it, but when they get stratified, you can get all kinds of weird data that look like that. If you look at production and decomposition, or production and respiration, which is really what we're measuring, you can see that they're fairly well coupled. So these are all different lakes that we've done work in, and so most of them follow pretty close to the one-to-one line. And you know, this shouldn't be too surprising, but what it tells us basically is that by a lot of mechanisms that we don't really understand, most systems are pretty in sync, you know, so they're mostly respiring what's being produced in the short term. So they're decomposing material that was produced yesterday or a few days ago, and they're dependent on that material in order to fuel respiration. So I guess, you know, it should make a lot of sense. If we look at annual balance in these systems, that zero line means that the system is neither heterotrophic or autotrophic. Okay, so that's the zero exchange with the atmosphere over the course of the year. You can see both, basically, our systems are pretty close to that. But I should caution, it's really hard to capture the dynamics, particularly at ice out and at ice on. Okay, and those could be important periods that we haven't captured very well. But for the most part, if you compare the clear and the turbid lakes, they look very similar. So in terms of the amount of material that's being produced in those systems, they're actually quite similar. And they're close to neutrality, but it can vary a lot in the season. Okay, so these bars, the production is in the gray, and respirations in the red, and then net productions in the blue. And you can see the numbers aren't huge, but you get net production in the summer, and that's kind of what you'd expect. Net losses in the spring, fall, and in the winter time. Okay. But again, the winter and the transition periods we haven't captured as well as I'd like to. So if you start to put it all together in terms of, like, in a given system, we can calculate essentially gross primary production. So the amount of CO2 that's taken up is on the order of 60 to 600 grams carbon per meter squared per year, which is similar to what other people have measured. Most lakes globally, from John Cole's work, tend to be more heterotrophic. So they move CO2 out into the atmosphere, probably because they collect a lot of material from the watershed as well, and that helps fuel that CO2 loss. But our lakes tend to be fairly neutral. So the fact that they're not negative sort of suggests that maybe they have more carbon to work with and more carbon to bury, which we know is true. And to pull it all together in a simple model in terms of the carbon cycle, this is what we think is going on. So we have, very similar to the pipe model I showed you before, we have about 100 to 200 grams of carbon per meter squared per year that probably comes in from terrestrial environments. The GPP, the production and respiration, are pretty close to being in balance. Burial is on the order of 80 to 150 grams per meter squared per year, and then they export about 50 to 80 grams. So what happens to this material is important because we don't really know. You know, is it eventually respired, or is it essentially equivalent to what's buried, you know, in terms of being removed from the system for a long period of time? But essentially what's going on is the internal parts of the system are pretty well balanced, and the reason we have export from these systems is because we also have a significant amount of import into those systems. And I think I put that in the context of the pipe model here, just to kind of draw these things together, but this is a simplified version, so 100 to 150 coming in. Efflux, in terms of the pipe model, in our systems, this efflux is relatively low. So most freshwater systems, if you compare it to the global measurement, the material that's coming out is quite a bit higher than what's being buried, but that's not really the case in our system. So in terms of comparing it to other freshwater systems, these wetlands are actually pretty significant in terms of carbon burial. Okay, I think that's it. happy to take any questions you might have. Yeah. I don't know what I did, but those are the collaborators. So yeah.
How do you convert this knowledge into a management scheme where counties or entities can target particular wetlands as being more important than others as a sink?
Well, I mean, I think that this, we know that we need wetlands for lots of reasons, right? It's a waterfowl issue, it's a water quality issue, but this is another motivation, I think, for preserving wetlands. And we certainly haven't done a very good job of it, but this is one of the motivations, I think, for that. We initially wanted to look at the issue of the turbid versus the clear, because to be honest, for all those reasons that I sort of discussed, like the different plant material, the productivity, we thought basically there would be a bias toward the macrophyte systems or the clear state systems to, because there would be more productivity and the plants degrade at a slower rate, and also less oxygen exposure. That I personally thought that we would see big differences in those two different management schemes. So the DNR is already managing for the clear water state. But that's motivated mostly by water quality issues and waterfowl issues. We thought that the carbon burial story would tell us the same thing, but it didn't. But I also think that the jury can still be a little bit out on that, just because our resolution in terms of burial in those systems is not very refined. And a lot of these systems can switch back and forth, so it's hard to know, like, how much of their life history they've been in the clear state and how much they've been in the turbid state, you know. But yet we're measuring the burial at a very crude scale. So, I don't know if that answers your question, but that's kind of where we're at. Christine?
Yeah, I was wondering if you could go back to that last picture of the ins and outs girls. And I don't know if I didn't quite understand the influx, but if a wetland system is in a watershed where it's getting, yeah, that one,
Oh.
a lot of input from
Terrestrial
all the
systems.
lateral material.
Yeah.
Is it going to be a system then that's exporting more material as well? So
That's a good question.
I guess why I'm asking that is because if that is the case, if you have a system that's getting a lot of input and therefore it's outputting a lot of carbon, would it be important or more important to make sure that that system is buffered in a way that reduces that input so that
Well,
the external exchange of carbon is more balanced, similar to the internal?
I mean, from a management perspective, I don't think we really know. So I can tell you a couple of my suspicions. So we've done some work. So I did a lot of work up at the Itasca Field Station for U of M. And so there's a lot of deeper lakes up there that I work in. And we see a very strong input signature from the terrestrial environment in the springtime in those systems. Actually, you see it in almost all lakes in Minnesota associated with combination of snow melt plus increased precipitation in the springtime and into the summer. And so that brings in a lot of material. But even in these wetlands, an average residence time for the water in these systems is, we think it's on the order of 1 to 3 years, depending on the wetland. So that's a long time. And so there can be a lot of processing of that material. It happens. And a lot of that terrestrial material gets metabolized in that period of time. So the material that comes out is not the same as the material that comes in. But what we found up at Itasca Lakes is that material that comes in seems to be very labile. It degrades fairly quickly, which is kind of counterintuitive because we mostly think of terrestrial materials being fairly recalcitrant to degradation processes. So a lot depends on, you know, how readily that material degrades that's coming out of the wetland system, and we don't really have a good handle on that at all. But that essentially, if it's very recalcitrant material, it could be equivalent to burial, right? It may be in the dissolved pool, but it could be equivalent to burial of carbon as well. But that's a really big and important question, I think. Yeah. Did you have one?
Yeah. So with methane being a much more potent greenhouse gas, this is focused on carbon. Have you guys done any methane analysis or have any suspicions? Because I've heard the pros and cons of wetlands, and they might sequester the carbon, but they the net export of methane can mean that. And I know it's very complicated. At least the research I've looked at is very complicated. Different times of years, different times of wetlands, and different climate conditions.
Yeah. So the bottom line is I don't know, but because we didn't focus on methane in this study, but the calculations I did based on what's been done in other places suggested that, you know, in terms of loss of CO2 into the atmosphere, that it's like, you know, 100 times greater CO2 than there is methane. Okay. So if methane is on the order of 20 times stronger greenhouse gas. So from that perspective, CO2 is a bigger deal. But it also means methane is not insignificant, okay? It's probably, and in our system, remember, our CO2 losses are not very high compared to a lot of the freshwater systems. So if methane is going to be important, it would be in these systems, but unfortunately we haven't measured it. But I just recently got some money from the National Science Foundation to do exactly that. We're not going to do it in these lakes, in these wetland systems, but we're going to do it in Lake Itasca. So we're going to put in a buoy, basically, that'll measure all those things like oxygens and conductivity, pH, you can do with a sonde, but in addition to that, we'll have thermistors throughout the water column, and we'll have sensors for CO2 in the water and CO2 out of the water. And I'm working with a soil scientist that has an eddy covariance system so that we can actually look at CO2 exchange across that interface. But in addition, we're going to measure methane. So we'll look at methane exchange across that interface as well. And then we'll also measure methane that's coming out of the water. So that'll be pretty exciting. We're going to do that in actually one wetland system. So we're going to do that out at Cedar Bog Lake, out of Cedar Creek, which is about as wetland as you can get. And then Lake Itasca, and then Elk Lake. So, yeah, so I'm pretty excited about that. Yeah.
Are the same processes going on that you described here going on in some of the really large peatlands of northern Minnesota?
Yeah, exactly. And you know, there's a lot of folks up in at the Marcel Experimental Forest that are working on that problem right now. And one of the things that they're looking at is CO2 and methane exchange, and they've been doing that for a long period of time. And there can be huge amounts of methane that come out of those fog systems. But more recently, they put in a system, they basically have these huge chambers that extend up, I don't know, like 12 meters or so, the size of trees. You know, systems that they're adding CO2 and warming up. So they have like a gradient in terms of adding CO2 and increasing the temperature. And they increase the temperature both in the soil as well as in the atmosphere over the soil. So, and the gradient for temperature goes from ambient up to, I think, +9 degrees C. So that's a big increase. And then the CO2 increase, I think, goes to 800 or 900 ppm. So they're basically, it's like hitting those systems with a sledgehammer, you know. So I went into a couple of them this last summer. They had just turned them on, and the ones with the high CO2 and the high temperature, it's like walking into a dry sauna or something. This felt horrid. And you could already see some of the plants dying. You know. Yeah, Christine?
I was curious, you mentioned that it's difficult to know which systems have flipped or if they flip back and forth from turbid to clear states. And I was wondering if any of the